All right. I think, maybe a lot of us are ready for lunch, but so I'll be quick. Really, I'm a computational biologist. I don't know why I'm here, so thank you so much, the organizer, to give me the opportunity. Really, for me, it's a tremendous learning opportunity. In this morning, I've learned supercomputing and MPI and Docker and fast I/O. I really want to use all of this into genomics. So that's what I really struggled in the past year. So I'm going to share this experience with you, and today is really, I changed the title a little bit because we're still very early in adopting Apache Spark in genomics. So I hope I can tell you a good story. So I like to start with something we all familiar ourselves. Actually, we're not that familiar about ourselves. So when we talk about I, we actually means we, because we together, our human cells plus 10 times those microbe cells living with us, form these super organisms to perform all the functions. And in later years, we start to learn that these microorganisms really impact our health, even our mind. So really, we are not only what we eat, and we're also, this microbiome drive us how to think. So I want to picture all the speakers like use this image. So when we're actually, this micro living with us are constantly exchanged with the environment. So hopefully that won't destroy your appetite for lunch. So microbes are really everywhere. So they form communities like what humans do. And on a human, there's actually only, we're talking about 1,000 different species cohabitat with us. And when we go out to the environment, like go to the cow, we're interested in the cow in DOE because cow represent a bioreactor for making biofuels. In their gut, there are about 6,000 different species living with them. And really, the champ is really, we go to the dirt. We just get above dirt, and they can contain up to 1 million different microbes. And we still don't know how many are there, as we don't have a technology to measure how many different kinds are there. And even we have this technology now recently, and now we are going to overcome a different challenge which is the big data challenge everybody's talking about. And to make things worse, over 90% of the species live in this environment. We have no idea of what they are and what they do because we have never seen them previously. So this is how we do. So this new topic is called metagenomics, really the study of the microbial communities. So the experimental and informatic approach started. So let me just figure out how to. So we started with the left up corner. So we collect the samples from human, from cow, from soil. We put them in a filter to get rid of this non-biological stuff, and then we're left a collection of all these different microbes. Then we harvest DNA from these microbes, and most of them are circles. I represent those DNAs using the circles. So each circle is a genome, and each color is a different species. So we're dealing with many different species, and each species have many different copies. And then, but when in terms of to determine their code or sequence, we don't have a technology that can read one genome at a time. So we have to break the genome into small pieces, and then we're using this massive parallel sequencing technology, or next-gen sequencing. They will end up a pile of these short reads, and those reads, I have the color in there, but we actually don't know which one is which color. They're really a mixture of these short pieces. And through this very challenging assembly informatic approach, hopefully we can recover the gene content, the content of each genome. And in a lot of time, we cannot do a perfect job. So I've used this dashed line to represent the reconstructed genomes. So I know most of you are computer scientists, so I'm trying to use an analogy to describe the complexity of this problem. So each genome is equivalent to a book, and metagenome, we're really talking about a library of books. So the assembly problem is for each of the genome, we shredded the book into random pieces, and the job is to put the pieces end to end together to recover the book. And for metagenome assembly, it's really the entire library of books are shredded into small pieces. And our job is really to try to reconstruct every book using the information, because each of the book have many copies and the sharing process is random. So we can use the overlap between those strains to stitch them back together. Yeah. Of course, there are two problems. First, there's noise during the shredding process. Some of the pieces, let's say you can say, contain water stains, so we cannot recognize the strains very faithfully anymore. So these are noise of the sequencing. And also, some of the books are more abundant, and some of the books are rare. They have only a few copies. So normally, we have to go very deep. We have to collect a lot of these short pieces to be able to see those rare copies of the book. So we are facing quite a few challenges. Scale is the first enemy. So commonly in metagenomic study, we're normally talking about 10 gigabases. I'm using the unit gigabases. They're roughly equivalent to one base is equivalent to 1 byte in storage. So most of the experiments we're dealing today are around about 10 gigabases. In 2015, we start to see projects, large human microbiome project, like they sequenced close to 2,000 humans in one experiment that generate half a terabase of sequence. And we finished a project in the cow. We end up getting 1 terabase of sequence. And for the soil, we haven't got enough data yet. And the leading researcher, James T.J. from Michigan State, postulated that we're going to need probably 20 petabases of data to be able to see the majority of the sequence in the soil. So I think the number might be a little bit deceiving here because that's just the number of the size of the storage to store the data. But when you try to analyze the data quickly explode. We did some early experiment. It turned out we need on average 100 times to 200 times of storage to be able to store all the intermediate data sets. So the other challenge is really the complexity. I mentioned about this contamination and those errors in the data. Those have to be cleaned out first. And also, when the data grows, the complexity also grows, because when you start to see the same strains over and over when you go very deep, because those abundant copies will show up more than others. And people are used to use this graph architecture, either using an overlap graph or a de Bruijn graph to try to solve the assembly problem. And when the graph getting very big, and it's very challenge to analyze. And also the graph traversal is always a problem, because some of the books share some of the text with others. So then they can form these overlapping graphs. So these repetitive elements and homologous genes and horizontal gene transfers, they represent similar text among different genomes. Since 2009, I started to think about this problem. And at that time, we have very few options compared to today's what do we have. But we know what kind of option solution we want. we, first of all, because the technology evolves so fast, and also science can't wait. So we actually put the easy to develop first, and partly because we are a genomic lab. It's very hard for us to retain very good software engineers, especially in the Bay Area. And also, we like the solution to be robust. It will, and many bioinformatic tools, if you ever touch them, and I think 50% of them just don't run in your environment. And we, of course, we like to scale to big data without changing the coding, ideally. And also, we like to be efficient. And we're not that like, you have to do it in minutes. We're perfect fine, if we have solution in hours, because, that part we can wait. So we have tried quite a few options. The first thing, when we were very naive, we said, okay, we have bigger data. We need a bigger computer to do that, right? So in 2009, we actually purchased a big memory cluster, and that's cost us like $1 million. So it does. We don't have to do anything to make the program run on the big computer. They just a bigger computer. And so, but they are pretty expensive, and we were only able to scale up to 100 gigabits of data, and then we cannot scale because the machine can become so big. And we also tried MPI solutions, and it is very fast, but it takes us a long time, and we're experienced software engineer to develop this. And we really cannot afford to do this. First, it's very expensive to maintain a team of very good engineers. And also, when the data change and the environment change, we have to change the code as well. So 3 or 4 years ago, we tried the Hadoop solution, and we really like that we can scale up data to one terabyte of data. But that solution is very slow. If you run it on Amazon, it ends up costing a lot of money. So a couple years ago, we started to see Spark. I think probably everybody of you know Spark. So this is a new scalable programming paradigm. So, okay, great. It can scale to big data. And also, it's much faster. It's claimed to be 100 times faster than Hadoop, so it's an efficient solution. And it has rich APIs, include Java, Scala, and Python. So we love that because it makes us easy to develop the software. Even as a computer biologist, I can program Python and Scala and work on our own problem. And also it has this interactive shell, which makes development much easy, and you can do something and see the change. So we don't know how robust this is yet, but we have heard so much that this is a very good solution. So in every way, Spark represents an ideal solution to us. So we decided to give it a try. So the goal. We want to develop an application based on Spark and solve our metagenome problem. So we would like to develop a clustering approach so we can reduce this metagenome problem into a single genome problem, right? So if we can collect all the pieces from a single book, from every book in the library, then the problem is much simplified. Then we can deal each of the book individually. And this also carries 2 other benefits, which we can parallel processing all the books. And also, we can afford to do individual optimization of each book, because each of the book may require a different parameter set for the best result. So the algorithm, the oversimplified algorithm is here. It contains 3 major steps. The first step, we're trying to identify which pieces are overlapping with each other. We do this in an approximate way. We count the number of substrings or chimeras here. that two pieces share, then we can overlap two pieces together. When we compute all the overlaps, and then we can form a read graph where in here the node are the reads and the edges are the number of chimeras they share. And then we can apply a graph partitioning algorithm like the label propagation algorithm I present here to try to figure out which pieces come from the same genome. Okay? And we use two test data set. The right one is a real car-room metagenome with sequence that has 1 terabase. So that this data set contains over 10,000 different species. It has very high repetitive elements, and there are very few known species. We get the data using a technology called Illumina sequencing, and the reads are very short. We get, for each of the pieces, we get 150 bases from each end, and we have a lot of them. We have 1.2 billions of short pieces. And this is not a good data set to validate whether the algorithm will work or not, because we don't know the answer. So that's why we use a human transcriptome data set. So this is not a real metagenome data set, but in every way, it simulates the complexity of the problem. And so, so in this data set, there's about 20,000 species. The repetitive content is medium, and we know pretty much almost all the species out there. So when we have a solution, we will be able to know the answer. And this data set is generated from a different technology called PacBio, and the read length are much longer. They can up to 20 kilobases, and there are about 2 million reads, and the total data size is about 8 gigabytes. So I'm not going to detail about how well this algorithm works. So this graph just show that in terms of purity for every cluster we form, also for the completeness for every molecule we should be clustered together, we got a very good result. You can see the histogram on the left side. And also for those impure clusters, we also look at why they are impure. And turns out most of the time is because we don't have sufficient data for this cluster. When we do have sufficient data, their purity and their completeness are much, they pretty much correlate with the amount of data we have for those genome or species. So I like to focus on the performance or scalability of this application we developed called Spark. It's Apache Spark-based read clustering. So we tested in 3 hardware and software environments. We were get a lot of help from a Chinese company called Huawei, and they have provisioned a cloud infrastructure in Germany called OpenTech cluster. So we got a small cluster on that environment with 20 nodes. There's 8 cores per node, 64 gigabytes of RAM, and running Hadoop 2.7.3 and Spark 2.11. So we also compared to a similar cluster on AWS EMR. And we recently also tried this on the on-demand Spark provision on HPC. We got a grant from Pittsburgh Supercomputing Center on their bridge system. We have a 8-node system there with comparable of number of cores and RAM to run the applications. So the first thing we ask, can the Spark application scale up to large data? So in this graph, we tried 20, 40, 60, 80, and 100 gigabytes of data. And for all the 3 steps, and also as well as the total time required for run the 3 steps, we get near linear scalability. So that's really exciting because with only 20 nodes with OTC, we can already run 100 gigabytes. So we are on our way to scale up to 1 terabytes. And if we increase the number of nodes, can we do our job much faster? And these two graphs, we tried on the 5 gigabytes of data and also on the 25 gigabytes of data. And in both cases, we see if we double amount of the nodes we use, we roughly takes half of the time to solve the clustering problem. And a lot of early adopters of Spark found the performance of Spark is very sensitive to the parameters like the default parallelism. It's really how small the data should be split onto the Spark cluster. So we tried the performance in at different parallelism levels. So this is at the log 10 level. So you can see over a very wide range, the performance didn't really change much. So we were very happy to see that we don't have to pay too much attention on tuning the Spark performance to get reasonable performance in a wide range of parameters. So we do observe very different performance result when we run the applications on different data sets. So for the human PacBio data set, because that data set is less complex, so we sequenced some of the books with many copies. So there's a lot of connections. And you can see especially the LPA stage takes a long time to partition the graph. And while in the Carl-Rumen data set, at this, I believe this the data set is 4 gigabytes. At 4 gigabytes, still you don't have a lot of connectivity, and that's why the edges part and also the LPA part does not take a long time to run. So we actually, if we want to project how much time we'll need for clustering 1 terabyte of Carl-Rumen data set, is probably more or less like the one we had now with the transcriptome data set on the left side. So we also compare the performance on the 2 cloud environments, on Huawei's OTC and Amazon's EMR, and compared to a on-demand Spark cluster running on Bridge. So it turned out for the same size of data, the time required to solve the problem is comparable. So we were so glad to see this piece of result because we have partnership with NERSC. We have grants on these HPC centers, and running applications on the supercomputing center is much cheaper than we run on the cloud. We do have a grant from Amazon, but we ran out that amount of credits very quickly. So I think I'm going to stop here just to summarize our early experience with Spark in genomics. I think the first most impressive thing is it's pretty easy to develop. The ability to write in Python and Scala and have an end-to-end solution in a matter of days instead of months versus MPI is a big factor for us. And it's pretty robust. We're able to run this many times, and we seldom have like the application quit. And we show that it can scale to big data. We haven't tried this terabytes or petabytes yet, but with 20 nodes, we already scale to 100G, so we are pretty happy already. And one of the most impressive feature for its flexibility. We developed it once. We can run it on the cloud. We can run it on HPC without any change of the coding. And we did have to change some of the configurations of the HPC cluster. But other than that, the application is just wrong. So how efficient this compared to Hadoop, we know is definitely at least 10 times or even more. We haven't compared to the Hadoop solution. And for MPI, we don't have a comparable solution in MPI yet. But I hope it's not that too slow than the MPI solution. And we got very good accuracy for those long reads, and we found that we need some optimization for the short reads. Yeah. So this is my slide. I really owe a lot of debt to our Spark team, who really like to take this challenge, because a lot of the time at the beginning we said, no, it's not possible, the scale of the problem. But really, the students, Li Zhen, Shi, and Xian Meng, they really team up and provide a solution and run with the help of all those founders, Huawei, Amazon, and Bridges, NERSC. We're able to develop this application in a very short period of time. We get a lot of help and advice from Lisa and Evan from NERSC, and Yongqing is here. When he worked at LBL, he helped a lot for us to adopt Apache Spark as well. So thank you for your attention. Any questions before the lunch? Okay.
Have you put anything in Docker containers? I mean, I'm biased.
Yeah. So I think I'm going to talk to you about that. I'm involved in a project called BioBoxes. It's a group of people from Germany, and also we're part of it too. Because running bioinformatic application is so challenging. 50% of the time, the application simply won't compile. And the other 40% of them, the things just won't run. And then the other 10% is probably very slow. Yeah. So I think the BioBoxes really takes a big step ahead. And we have put almost all the common tools we're using into Docker containers. Let's talk. I think Huawei is just experimenting. We haven't compared the price because for us, everything is free for now. But they claim I think for them to gain some users, it will be crucial for them to be cheaper than Amazon, because Amazon cloud is so easy to use. And their cloud, you still have to do some configurations to be able to use it. Yeah. All right. Yeah, thank you again.